All right, everybody. Module 4A, Lab 7, Cellular Biology. This is the basis of all biology, okay? Here we go. All right, what about cell theory timeline? It started with some people who did some inventing. According to your book, it turns out that the invention of the microscope happened because of some people selling fabrics, wanted to look at it a little more closely. They would use something similar to a magnifying glass, and someone figured out if you put two magnifying glasses together in a certain way, it even makes it more. You know, my kids laugh at me if I put my reading glasses on, then I put a second pair of reading glasses on. It definitely magnifies it even more. It really works. It's a double lens thing, okay? So somebody had figured that out. I don't remember those guys, because the famous people would be Robert Hooke and Antoine von Leeuwenhoek. Okay, those are the two guys that really, really took the microscope to levels where they could see creatures in Hooke's case, it would have been plants. We're going to try to draw something today. We're going to try to make a slide and draw it today. And then for Leeuwenhoek, it would have been microorganisms like bacteria and things like this. And then came along Schleiden and Swan. They were kind of friends, one working with plants, one working with animals. And they came up with the idea that everything, all living things, are made up of these things called cells. It was Hooke who came up with the name cells, because it looked like little places where monks lived in a cell, like a jail cell, really. That's how he named it that. And then we're going to see after the fact that we're Francesco Redi, and he's the one that disproved spontaneous generation, and then along with Louis Pasteur. I have a couple other guys here, Robert Brown. You'll read about him in your book, and Louis Pasteur at the end there. So the progress of cell theory, there's Hooke, here's Schleiden and Schwann, and a guy called Rudolf Virchow. Let's watch a little video on it. Oops, sorry. Oh yeah, this is overview of cell theory. Good, I had it at the end.
Cells are the smallest living units of an organism. All cells have three things in common, no matter what type of cell they are. All cells have a cell membrane, which separates the inside of the cell from its environment, cytoplasm, which is a jelly-like fluid, and DNA, which is the cell's genetic material. There are two broad categories of cells. The first category is eukaryotic cells. They have organelles, which include the nucleus and other special parts. Eukaryotic cells are more advanced complex cells, such as those found in plants and animals. The second category is prokaryotic cells. They don't have a nucleus or membrane-enclosed organelles. They do have genetic material, but it's not contained within a nucleus. Prokaryotic cells are always one-celled or unicellular organisms, such as bacteria. So, what are organelles? Organelle means little organ. Organelles are the specialized parts of a cell that have unique jobs to perform. Let's start with the nucleus, the control center of the cell. The nucleus contains DNA, or genetic material. DNA dictates what the cell is going to do and how it's going to do it. Chromatin is the tangled, spread-out form of DNA found inside the nuclear membrane. When a cell is ready to divide, DNA condenses into structures known as chromosomes. The nucleus also contains a nucleolus, which is a structure where ribosomes are made. After ribosomes leave the nucleus, they will have the important job of synthesizing or making proteins. Outside the nucleus, the ribosomes and the rest of the organelles float around in cytoplasm, which is the jelly-like substance. Ribosomes may wander freely within the cytoplasm or attach to the endoplasmic reticulum, sometimes abbreviated as ER. There are two types of ER. Rough ER has ribosomes attached to it. And smooth ER doesn't have ribosomes attached to it. The endoplasmic reticulum is a membrane-enclosed passageway for transporting materials, such as the proteins synthesized by ribosomes. Proteins and other materials emerge from the endoplasmic reticulum in small vesicles, where the Golgi apparatus, sometimes called the Golgi body, receives them. As proteins move through the Golgi body, they're customized into forms that the cell can use. The Golgi body does this by folding the proteins into usable shapes, or adding other materials onto them, such as lipids or carbohydrates. Vacuoles are sac-like structures that store different materials. Here, in this plant cell, the central vacuole stores water. Going back to the animal cell, you will see an organelle called a lysosome. Lysosomes are the garbage collectors that take in damaged or worn-out cell parts. They are filled with enzymes that break down this cellular debris. The mitochondrion is an organelle that is the powerhouse for both animal and plant cells. During a process called cellular respiration, the mitochondria make ATP molecules that provide the energy for all of the cell's activities. Cells that need more energy have more mitochondria. Meanwhile, the cell maintains its shape through a cytoskeleton. The cytoskeleton includes the thread-like microfilaments, which are made of protein, and microtubules, which are thin, hollow tubes. Some organisms, such as plants, that are photoautotrophic, meaning they capture sunlight for energy, have cells with an organelle called a chloroplast. The chloroplast is where photosynthesis happens. It's green because it has a green pigment called chlorophyll. Plant cells also have a cell wall outside of their cell membranes that shape, support, and protect the plant cell. Animal cells never have a cell wall. There are many other unique structures that only some cells have. Here are just a few. In humans, for example, the respiratory tract is lined with cells that have cilia. These are microscopic hair-like projections that can move in waves. This feature helps trap inhaled particles in the air and expels them when you cough. Another unique feature in some cells is flagella. Some bacteria have flagella. A flagellum is like a little tail that can help a cell move or propel itself. The only human cell that has a flagellum is a sperm cell. In summary, remember, eukaryotic cells are plant and animal cells with a nucleus and membrane-enclosed organelles, while prokaryotic cells are unicellular organisms without these things. All cells have a cell membrane, cytoplasm, and genetic material. And even though only plant cells have chloroplasts, both plant and animal cells have mitochondria.
Okay, we're going to cover each one of these now individually. The cell theory, I showed you the infographic earlier of the different people. Here's what has come up. You have a box in there. All living things consist of cells. They're either unicellular or multicellular. The cell is the basic unit of structure and function of life. And cells always arise from existing cells. Life must come from life. We do not get living things from non-living things. So these are rules of biology. So if somebody believes that all of life came out of nothing, they've just broken the rules of biology. All life comes from life. They're wrong, yeah. So this is mainly the work, the combined work of Schleiden and Schwann. but working from the other people like Hooke and von Leeuwenhoek and those guys.
So Italy Max.
Try to write down bullet points so you don't have to write down every word. It gets a little long that way.
Time.
Okay, I'm gonna move on. Typically, we draw an animal cell as a circle. Remember, we're looking at these would be eukaryotic cells. And so we draw an animal cell as a circle and a plant cell as geometric, usually a square. Here it's drawn as a little different in there. So cytology is the study of cells. It's underlined, I think that means there's a blank for it. Cytology is the study of cells. When I worked at Brackenridge Hospital, I worked in the lab area, and one of the lab section was called cytology, and we would make slides and look at cells under the microscope.
Nice.
That was the fun stuff. Yeah. We're looking at just some skin cells or something right there. There are 12 specific characteristics of cells. First of all, they come in different shapes. So while we usually draw an animal cell as circular, they can be really weird shaped, like that one there, which is a neuron, which is a brain cell. They're very strangely shaped. We have our bacteria shapes. These are cells of the kingdom Monera, but I think we're going to be looking at plant and animal now. Here's white blood cells up here, red blood cells. Okay, so the first word is ingestion. Ingestion. I believe, are you doing a matching or putting a...
There's boxes.
Serious?
Wow.
So write ingestion. Write the word ingestion.
There's 12 boxes.
The ability to take in nutrients, including absorption into the cell.
Do you write that? Or
You're
should I
just
just-
writing ingestion. I think at the end we're going to come back and do matching. Does that sound right? What is on this page? Is there matching on this side?
Oh, yeah.
Yeah, so just write down ingestion is number 1. We can see in the picture right here, these purple things are being ingested by the cell and closed in, and it starts outside the cell, ends up inside the
I have a
cell.
question. Do we go from left to right, or we go
Doesn't
up and
matter.
down?
They're not in any sort of order.
I think it matters. Okay.
No order. Number 2, respiration. Respiration takes place in this structure right here called the mitochondria. You saw
Powerhouse of the
that?
cell.
It's the powerhouse of the cell. Power is produced through the process of respiration. The ability to release energy from the breakdown of food molecules. It's happening in the mitochondria. So if you want to put respiration and put in parentheses mitochondria, you can do that if you like. But we'll get back to it. We'll come back to it. That's the really cool mitochondria. We're going to do this Krebs cycle in a different module this year. It's one of those things that strikes fear into the heart of every undergraduate studying the Krebs cycle.
Why does it strike fear?
Because it's complicated. Massively complicated.
It's pretty easy. No, it's not.
Yeah, it's not easy. Homeostasis, number 3, homeostasis. The ability to maintain internal stability. It's also known as regulation. All right, and we're looking at a very strangely shaped cell right there. That's called a neuron. It's the cell that sends electric signals from your brain to your body. And it goes through a complex cascade of events called the sodium-potassium pump just to send a signal down, to make you move your eyes to look up there and look down. All of that has to happen in all those little cells right there. It's quite complex and quite miraculous. Number 4, secretion.
Is that saliva? No.
Well, it's a secretion, sure. Ability to release biosynthesized substances, and that usually comes from the Golgi apparatus or the Golgi body. Next time when we get together, we're going to make a giant model of a cell. Today we're going to look at them under the microscope. All right, so a secretory vesicle came from the Golgi apparatus. It's being sent out. For instance, my pancreas is making a special chemical called insulin, and it through the Golgi body, it sends it out of the cell to the rest of the body that needs insulin to put glucose into my cells. Question.
Does that mean when people take their pancreas out, that means we don't get glucose anymore?
You're dead if you take your pancreas out. You cannot live without your pancreas.
But
Yeah.
do we need our appendix?
You do not need your appendix.
Oh, that's the word.
Is that what you're thinking of?
Yeah.
That's part of your
Have we
digestive
ever
system.
needed our appendix?
Actually, I would say if you have the opportunity to keep your appendix, keep it.
So like, what's wrong if you take it out?
It removes some lymphatic tissue from your body, makes you a
Exactly.
little more subject to disease.
But you could live without it.
You can live without it, but you cannot live without your pancreas. All right, number 5, egestion. Notice how similar that sounds to ingestion. This is egestion.
Is that thrown up?
All right, this is the ability to remove non-soluble, undigested waste from the cell. So egestion is a waste removal
So it's
system.
like throwing up through your cells?
I guess so. Yeah, here we go. Our lysosome mixes with it and it sends it out, gets rid of it.
Oh, can I throw my
Uses
gun
what
away?
it can, gets rid of the other. What's the question?
Can I throw my gun away?
Certainly.
So he's
All right,
doing
number 6,
digestion.
movement.
So-
Number 6, movement, the ability to move. Motion. All right, what we see in this picture right here is a white blood cell moving through other blood cells. It's see that little thing right there? It's chasing? That little tiny thing? Oh, it got it at the end. That was cool. Check it out. It's going to chase that little guy. Then it's going to gobble it up. It's chasing a bacterium. Then it gobbles it and eats it and gets rid of it. Awesome.
Wait.
That's a white blood cell.
All right now.
Movement. Just write down movement, the ability to move. Number 7, digestion. All right, the ability to break down nutrients by hydrolysis or enzyme action. My food comes in, I break it down into amino acids if I'm making a protein, or I break it down into simple sugars if I'm using it as a carbohydrate, or if I'm making a fatty acid, I'll store it as fat, as a lipid, right? I can make those macronutrients right over there. A, B, and C are the food. D, E, and F are the things that are being made. We're going to say protein, carbohydrate, and fat. Number 8, transport. The ability to distribute or circulate molecules from one cell to another. Next week we will talk detail about how this happens, and we'll do a really fun classic experiment. But that's next week. If you see there's a little wall right here with holes that are a certain size, big things can't get through those holes. It's like a screen on a window.
Or like
A screen on a window allows air in, but not mosquitoes.
Or like the word strainer.
A strainer. There you go.
Swiss
Number
cheese.
9, synthesis. Synthesis. The ability to combine simple compounds into more complex compounds, oftentimes called biosynthesis because it has to do with putting things together in a living creature, therefore it's bio. Okay, and the synthesis right here, what's happening in this picture is that green thing is a ribosome, and this code is coming into it called mRNA. We'll have a whole unit about that, a whole module, and that's another one of those make you nervous kind of modules when we talk about protein synthesis. It's making a protein right here. What are those little balls? Do you remember?
Oh,
Long chains of
proteins?
amino acids. Amino acids.
We're really close.
Number 10, excretion. Not secretion, but excretion. The ability to remove soluble metabolic waste from the cell.
So it's like throwing
Remember, you're just writing down the word excretion here.
So like anything in here that starts with an E is basically just thrown out.
Getting rid of
It's
some
going
X, like exit, right? Like exit. Oh, 11, irritability. The ability to respond to stimuli. If I punch you in the face, you'll punch me back. Yep. If you touch something hot, you'll move your hand quickly away from it.
I don't want to.
I don't know what this water's doing for my eyeball. Oh, I see the water. My sensory neuron sends a message to my arm to lift the water glass and drink it. All that happens with the sodium potassium pump in each one of those little neuron cells that are spreading that signal all the way through your body. Number 12, reproduction, the ability to produce more cells. So that should be all 12, yes? 12 things? And we'll do some matching at the end, I believe. What we're looking at in this picture right here is a single cell that is formed into 2, then 4. So it's going to look something like this, right? Ingestion, respiration, homeostasis, secretion, egestion, movement, digestion, transport, synthesis, excretion, irritability, and reproduction.
Yes, ma'am. But I didn't write all the other things.
I know, you
Other
were just
things.
supposed to write the word down because I believe we're doing a matching.
Yeah.
later.
Where?
A matching, but not with.
Yeah, but not with
They're
that.
different words?
Yes, ma'am.
I think we can remember these.
Yeah, I think that's easily memorable.
I don't know.
I think it'll be fun.
Oh, shit.
I don't know if we have to know such detail.
Fill in next quizzes. Those are good.
All right. Now we have blanks to fill in? Or are we on matching now?
Do you have some blanks to fill in, ma'am?
Oh.
They're right there. Yeah, those 2 words right there.
Structure and function.
All right, cellular organelles. We're gonna look there on that picture on the left. You're gonna see things numbered, 1, 2, 3, 4. Where's 5? 5, 6. Okay. An organelle describes a tiny cellular structure that carries out specific functions necessary for the cell to survive. Does anybody know what number 1 is up there?
Nucleus.
Number 2 is the nucleus, so the thing inside the nucleus. In the video they called it a nucleolus.
Number 9's the
Nucleolus, but I
mitochondria.
call it a nucleolus.
Number 9's the mitochondria.
Number 9 is mitochondria. Did you say anacondia?
An accident.
Mitochondria.
It's hard for the stuff.
3 is the rough endoplasmic reticulum. 4 looks like a vacuole. 5 is the other endoplasmic reticulum. Golgi body. Oh, here's smooth endoplasmic reticulum. I'm not sure what 3 is asking me then. Vesicle. Mitochondria. Lysosome. Centrioles. We'll get to it.
You got that Wi-Fi.
All right, here we go. 2 types of cells. Prokaryotic cells.
Where do you write this at?
No nucleus. Is there not a box for it?
There's
There is.
box.
Right there.
When I put it in a box, usually it's because it's in a box in your book. Okay? Prokaryotic cells have no nucleus. Remember, I'll accept that. No nucleus. Eukaryotic cells have a nucleus. Also, other membrane-bound organelles like mitochondria, ribosomes, endoplasmic reticulum, Golgi body, all those things.
Flowers. Maybe to you.
Too bad. I ain't reading.
Pink.
All right, structures that both types of cells have in common. They all have DNA. All living cells have DNA. The DNA is the code for life. They all have cytoplasm. They all have a plasma membrane. And they all have ribosomes. So you could argue that ribosome is a membrane-bound organelle, but it's not very large. But the ribosome is responsible for something. It's responsible for taking a code from the DNA and making a protein out of it. So that's how everything is going to exist, by getting a protein made.
If we take any one of those out,
It's
life
no longer
can't
alive.
exist.
Right. Right. Yeah, if there's no cytoplasm, it dries up. If there's no membrane, it just leaks out everywhere. No ribosomes, it can't make protein, and no DNA, there's no code to tell it what to do. All right, draw the prokaryotic cell at the bottom left on page 32.
Easy.
Bottom left
Is that what I said? Is that where it is? It's at the top?
of 32.
Is it 32? What's next? 36. 36, sorry.
Oh.
They changed the teacher book. Well, they changed your book and didn't change the
How
teacher
do
book.
we draw a circle with a tail?
Draw like a, bean shape
Are you supposed to be
there.
on the inside? Like
Yep,
the
and
white
anytime I ask you to draw something, I insist that you also will label it. I assume you understand that I want you to label it.
Cannot see it.
You can't see it?
No,
What are we
I don't have my
labeling?
glasses on.
Well,
Drawing the top,
maybe you
bottom,
should get
left.
your glasses.
I left my
Did you
glasses
say bottom or top?
that room.
I thought you said the bottom.
Wherever the box is. It used to be at the bottom.
Oh, it's not.
It's not anymore.
Actually, just do K, just
So remember, there's no nucleus, so we just have DNA floating around in there.
I'll just read the video later and put it there. Put the labels.
The pictures in your book as well. The blue stuff is cytoplasm.
I don't have
Ribosomes. Then it's got these things. We have a plasma membrane. It has a cell wall. It says here capsule. Not all prokaryotes have capsules. Some of the most notorious ones do. It makes them sticky. For instance, the stuff that causes strep throat, it's got a sticky capsule that'll stick to your throat, and that's how it makes you sick.
Disgusting.
Not every cell has a flagellum, but a a lot of them do. They give some motility. They can swim through water. Things like cholera, stuff like that, have flagella. And then these pili, those are also part of the capsule that make them sticky. They also can trade genetic information that way, but we'll get to that in a different module. Module.
Pill. That's a new thing. The red thing. Does it not? It looks look good. It's like I think we're good blind, Eddie. Frankly, honestly.
All right, everybody good?
Almost. Toddler. Looks like a toddler. It's not. Eddie, it looks like your brain cells. Roast. Listen to that shit. How was that, Eddie? Holy shit. What'd you say? It's body language. It's fine, Eddie. It looks like, Eddie, it's fine. You see
All
how
right,
it's
moving on.
Even if you're just studying, a lot of work is going on in your cells.
That's crazy.
Let's zoom down and check it out. We've arrived in the space between two cells. A sticky coat called the extracellular matrix holds the cells together.
Cytoplasm.
Each cell is surrounded by a flexible plasma membrane with an incredible number of projections, docking stations, and channels. Let's dive into one of these channels to enter a cell. Whoa, look at this place. These girders and cables make up the cytoskeleton, the structural framework of the cell. They also serve as tracks for transporting cargo from one place to another. All this activity in the cell requires energy in the form of ATP molecules, which are made here in the mitochondrion. Notice the outer membrane and the inner membrane with its numerous infoldings. Many of the molecules involved in making ATP are built into the inner membrane. All those folds increase the inner surface area, enabling more ATP to be made. Moving towards the nucleus, we pass by layers of internal membranes. The nucleus is enclosed by a double membrane called the nuclear envelope. Let's enter the nucleus through a pore. The nucleus houses the genetic material of the cell, DNA, which carries the blueprints for making the cell's proteins. Almost 2 meters of DNA is crammed inside the nucleus. How does it all fit? The DNA is wrapped around proteins like thread wrapped around spools. Look, this section of the DNA has unwound, and a different protein has attached to the DNA. DNA is being used as a template to make mRNA. mRNA molecules travel from the nucleus to the cytoplasm, carrying the instructions for making specific proteins. In the cytoplasm, a ribosome clamps onto a strand of mRNA. The ribosome ratchets along the mRNA, building a new protein. Some proteins stay in the cytoplasm. Others, like this one, are processed in special compartments within the cell.
So all this is happening right now?
Mm-hmm.
Protein processing and certain other metabolic activities occur in the endomembrane system, the cell's network of internal membranes. The endoplasmic reticulum, or ER, is part of the endomembrane system. There are 2 types of ER, rough and smooth. Rough ER is covered with ribosomes. Smooth ER lacks ribosomes. Lipids are made in the smooth ER. Let's go inside the rough ER. Note that you can still see the ribosome on the outside surface. The ribosome is manufacturing a new protein, which continues to grow inside the ER. Completed proteins move to the edge of the rough ER and depart in a vesicle that buds off from the ER membrane. Some vesicles fuse with the Golgi apparatus, another component of the endomembrane system. In the Golgi, proteins undergo further processing. Finished proteins are then packaged in vesicles that pinch off from the Golgi and are transported along cytoskeleton tracks. Some vesicles bind with the plasma membrane, secreting their contents outside the cell. Other vesicles, called lysosomes, contain digestive enzymes. Here, a lysosome fuses with a worn-out mitochondrion and breaks it down. Each of the trillions of cells in your body is a dynamo of activity, requiring millions of ATPs every minute. But most people are unaware of all this activity in their cells.
All right. So when we look at that, we're going to see all those different organelles, right? So we're going to see here plasma membrane, cytoplasm nucleus, endoplasmic reticulum, Golgi apparatus, all these. And I think we're going to go through individually. Those are our matchings, right? Those words look right? Okay. All right. So check off each structure in your course supplement when we reference it using the images. Okay. So we're looking at animal cell. So we're going to take each one individually, and are you drawing a line or putting a letter somewhere?
It said check off,
Oh, check it off? That's all you're doing?
I guess.
Okay. All right. So the first thing is the cell wall. Now remember, animal cells do not have a cell wall, only plant cells and some bacteria cells.
I don't
Yes.
see cell wall on here.
It's not on there?
No.
It's a rigid structure outside certain cells, plants and bacteria,
All it is a bunch of words, there's no definition.
usually made of pectin and cellulose.
There's no definition, it's just words.
All right, I guess you just have to check.
But there's no cell wall.
Okay, then leave it.
There's no.
Well, I started with it probably. All right, but I still need you to know what a cell wall is. Where do we find it? Plants and some bacteria. Okay? Function for protection, but it's permeable. It's got giant holes in it. Is plasma membrane on there?
Yep.
So all you have to do is check it.
Do
All right, well, listen to what I say.
That's the hardest task I've ever done before.
It's got a double layer of phospholipids, it's magic, that make up the outer membrane of every cell, plant, and animal. It's semi-permeable. We'll work with that next week. We'll get into more detail about semi-permeable membranes. That means some things can go in and some things can't. It's like the screen on the window, okay? It controls what comes in and what goes out of the cell. The phospholipid bilayer is an elegant design that protects the cell while tightly regulating hydrophobic and hydrophilic substances. Hydrophobic means it's oily.
afraid of water.
It's oily, yeah. Hydrophilic means it'll dissolve in water. A substance needed or rejected by the cell. So it kind of looks like this. All right, we have these double layer here, a phospholipid bilayer. Here's the top one and the bottom one. The lipids on the bottom, that's fat, remember. So it is oily. And then inside we have these phosphate parts that attract water. And then we'll have all these markers on there. This one is a channel to allow things in. We'll look at this more next time. Another protein molecule allowing things in.
But what are those black things in between them?
Those are the phosphates of the lipid phosphate molecule. So it's like got a head with a little tail coming down. We'll look at that with more detail next time. Cytoplasm, a jelly-like fluid inside the cells. Go ahead and check it. Cytoplasm. While it holds all the organelles in place, it also contains ions, which are atoms with an imbalance of electrons and protons. It produces an overall electric charge of an atom, or it's neutral, depending. Cytoplasm is constantly moving around in a process called cytoplasmic streaming, moving ions to places where they're needed. I want you to think of cytoplasm a little bit like a really elaborate Jell-O salad.
Jello salad.
Yeah, we got a bunch of fruit suspended in jello.
I never heard that in my
I've
life.
never heard of
Does
that
that look like
before.
ice?
Now I am hungry.
Terrible.
Y'all ever had jello with fruit in it?
It does look kind of
I have.
gross.
Sometimes in order for the fruit to be suspended all the way through it, you have to do it in stages, because if you don't, all the fruit will float to the top. So if you do part of it and let it cool off, then you put the next part in and put it in and let it cool off. That's how you can also have jello with different colors. You have to do it in stages.
Why is that
Alright,
grape so brown?
So why these are the organelles, the fruity organelles, the jello holding it in place would be the cytoplasm. Alright, we're gonna watch a little video on cytoplasmic streaming.
Is that a word too? No.
I don't think it is.
This video shows cytoplasmic streaming in leaf cells. What we're looking at here are the cells of the parenchyma of a leaf. The dark green ovals inside the cells are chloroplasts. Notice how they stay towards the edges of the cells. That's because they're trying to gather light, and the best view of the light is going to be at the edge of the cells. To further maximize their exposure to light, cytoplasmic streaming causes the chloroplasts to travel around the cell. That way, no matter where light is shining brightest on the cell, the chloroplasts are exposed to it.
Alright, mitochondria.
Alright, up to the cell.
Small membrane-bound organelle with specialized internal structure. We know that it's the powerhouse of the cell. This is where energy is made. If you think of the cell like a city, this is the energy department, right? The electricity department. So it makes something called ATP. ATP stands for adenosine triphosphate. That is a molecule with a lot of energy in it. Yes.
So it also called the mitochondrion?
That's singular, yes. Mitochondrion is one, mitochondria is many. It's typically your cells have more than one. But yes, that is correct. Notice its structure, it's got some pretty elaborate structure over there. When we talk about respiration, we'll talk about it with detail. Here it is. Check it out. We got its own ribosomes. It's also got its own DNA. It's a separate DNA. And it belongs to it alone.
What's that?
Very bizarre. Some people think that it used to be an organism and our cells incorporated it to make it be the energy producer for our cells. But how, it doesn't make sense because our cells, how would they have done it without it? They needed energy, so I think it was there the whole time. All right, here's a little.
Our body is made up of trillions of cells. They all require energy to function. This energy is created within our cells, in the mitochondria. Here, food is converted into chemical energy called ATP. ATP is released by the mitochondria so cells can use it. Mitochondria consist of two membranes, an outer membrane, separating it from the cytosol, and an inner membrane, surrounding the so-called matrix. The area between these membranes is called the intermembrane space. ATP is generated at the inner membrane of mitochondria by an efficient mechanism called oxidative phosphorylation, involving several membrane protein complexes. Nutrients provide high-energy electrons in the form of NADH, which are used by the protein complexes to pump protons from the matrix to the intermembrane space. This continuous pumping creates a proton gradient, where the positively charged protons are attracted to the more negative matrix. When the protons re-enter the matrix through the ATP synthase protein complex, they catalyze the production of ATP.
That music was going to make me go to sleep.
Well, he looks like he's falling asleep.
This is a picture of some mitochondria very, very close up. It tells us what? Nanometers right there. So and you can see that internal structure. And that internal structure is where a lot of the action takes place. Our next thing's called a lysosome. A lysosome. Okay. It is an organelle that excretes enzymes aiding in intracellular protection. In animal cells, lysosomes break down proteins and sugars, and they can release bacteria-killing enzymes. So it's for protection. Did you check that one off?
Yes, ma'am, I did.
Okay.
First, actually.
Ribosomes. floating around in the cytoplasm or attached to the endoplasmic reticulum called the rough endoplasmic reticulum. They are the cell's protein factories. Okay, they're making protein. We call that protein synthesis. Is taking place in the ribosomes.
Can we call them ribs?
Endoplasmic reticulum. That's a folded membrane
Is it the rough one or the smooth
next
one?
to, I think we'll maybe talk, do you have both of them on there?
Yeah.
Okay.
Just check
Let's see
one.
what it says. The smooth ER supports nuclear materials, the nucleus itself, makes lipids and hormones. The rough ER aids in the production of proteins and transfers new proteins. That means you can check them both off.
Yes, ma'am.
Smooth and rough. Remember, rough just means it has ribosomes on it. Plastids. Plastids can be found, most of the time we find these in plants. Leucoplast, leuco means white. Where do we see that? Right here, and that can be turned into these other plastids. Chromoplasts store pigments for photosynthesis, like chloroplast and chromoplast, right here. That gives color to the cells.
Yeah.
Amyloplast, elioplast, proteinoplast, depending on where we find them. These up here are just the stem cells for these other ones up here. They'll turn into those things. Right here we're looking at some plant cells, and those little green things in there are chloroplasts. And inside the chloroplasts are chlorophyll molecules,
Are they
which
actually
happen
green?
to be green. That's why plants are green.
The
When we get
green
into
man
our-
running around in there.
No green men,
My question
sorry.
is like, how come they are green? Like, how does
God picked green.
and how come some other plants are like different colors but they're so green?
All plants are green, so they can go through photosynthesis. They need chlorophyll. They have to have
But
chlorophyll.
then why are some plants like red?
They have other pigments in there, but they still have green.
What about blue?
When we get to botany, we'll cover this in
Where
detail.
have you seen
A
that?
little too much detail, in fact.
Blue bonnets?
Vacuoles and vesicles. Are those together or separate on your chart? Vacuoles and vesicles.
I got him.
The central vacuole in plant cells
You found
is a large structure that maintains fluid balance, something called turgor pressure. We'll get to that. Waste vacuoles contain waste products. Phagocytic vacuoles contain substances the cell has engulfed, like we saw that white cell eating that bacteria because it's a phagocytic vacuole. Similar vacuoles allow for penocytosis and secretion. We'll get to that. Next, we have the Golgi body or the Golgi apparatus. It's a stack of membrane-bound organelles, all stacked up like a stack of pancakes. It stores proteins and lipids and changes them. It folds them, maybe adds a few things to it. When needed elsewhere, the Golgi bodies will package these items, proteins and lipids, guys, and send those out via secretion and excretion. All right, the nucleus, hooray. The nucleus is in the center of most of the cells, covered in a double membrane, and the DNA's in there. When I'm asking you what's in the nucleus, I'm almost always asking you to tell me the DNA's in the nucleus, okay? DNA. The function is reproduction, cell division, also to send out the code for the construction of new proteins. It tells the ribosomes what it
Has
wants.
anybody ever like harvest one from somebody? A nucleus?
Sure, they're in every living cell of yours. If you give a blood sample, well, red blood cells don't have one. They don't have a nucleus because they don't live very long.
They're
But white blood cells do have giant ones.
Oh.
Hmm?
I said they're failures.
Well, they actually perform a really important job. They carry oxygen, and they don't have to do anything else. They don't divide. They can't go through mitosis. Cytoskeleton or microtubules, that is giving structure to the cell. They're mostly like fibers. I always kind of think of like little spaghetti strands in there. Microtubules aid in their shape and the motility of cells. They hold the cell's shape together. All right, and here's a picture of those special microtubules called centrioles, found mostly in animal cells. We don't know exactly how they work. Okay, so here we are, we're studying things and we think we know so much. We're not exactly sure what the microtubules do, the centrioles do, but they have something to do with cellular division. That's all we know. All right, they facilitate cellular division. That means they get it started.
What is that stuff in between them?
Those are called lamellae. They're like little tissue that holds those little tubes together, and they actually will turn into two and then pull the cell apart.
Are they actually clear?
I don't know. That's probably an artist's rendition of it. Microfilaments, fine thread-like proteins found in the cell cytoskeleton. They help with movement and support. Right here, here's a flagellum it's showing right there. Maybe it's part of that.
Flagellum?
How many blanks do we have? Quite a few?
Like, 1, 2,
Not that
3,
Not that
4,
many.
4 left?
Chromoplasts.
I said 6.
Plastids containing yellow, orange, or red pigments used in photosynthesis. This would be like, for instance, have you ever had a carrot? They're orange.
No.
Okay?
Carrot,
They have
actually.
chromoplasts in there.
I've actually never eaten a carrot.
Never?
So,
Why not?
I don't know.
They're delicious.
They are delicious.
Chloroplasts, they're green, of course. Plastids containing a green pigment called chlorophyll used in photosynthesis. Already checked that one off, yes.
Check them all off.
Do my legs count as flagellum?
No,
Okay.
they do not.
That's my mid-children sugar.
Peroxisome, a cell organelle containing enzymes that catalyze the decomposition of fatty acids and hydrogen peroxide, similar to lysosomes.
H2O2.
That's hydrogen peroxide.
Shows.
Middle lamella. These are the lamellae. This kind of like what we saw in those microtubules right there. Thin film between cell walls of adjacent plant cells. They're made of something called pectin. Pectin's a little bit like jelly. If you've ever made jelly before, you add pectin to it. Literally, it's called pectin.
Yep.
So the middle lamella is right here. Primary cell wall here. It kind of is. Let's say it's between the cells. Oh, this is interactive cell website demo here. Let me see if it works. It's just gonna, if you wanted to go to this, you can see the nucleus and it circles the nucleus. Then you move it down to the next thing, the nucleolus, and it circles that. So we're not gonna go spend much time on this one. Do you have in your book a place to draw an animal cell and a plant cell? Is that what's next? Okay. So go ahead and here's your animal cell you're gonna draw in the box for animal cell.
Wait, which one's which?
Is it?
There are two.
There's 2 boxes?
Are there 2 boxes?
Yeah, from that conversation.
Okay, maybe you don't have to. Hold on, guys. Hold on.
Hold on, there's.
We're doing this next time. Oh, check this out. Now, the cell structure. Okay, here's what I'm gonna tell you about this thing right here in your book. This is homework. Guys, right here in your book, this is homework. That means when I take this book up at Christmas time, I want to see stuff in there. You're gonna fill
So
in
what
what
is-
those
what's the-
things are. You see that? Yeah. So make sure you do that. So don't draw this then in that little box at the bottom of page 36. What we're gonna do instead is let's get started on our lab, okay?
What's this
All right,
one? Is
but
this
animal
one?
cell, really quick, animal cell and plant cells are similar, except animal cells do not have a cell wall, plant cells do. All right, and notice the large central vacuole. In some ways, plant cells are more complex than animal cells, but at the same time, animals are more complex than plants, right?
You said don't draw it, right?
Don't draw it, we're gonna leave those open for something else.
Yes, ma'am.
We're gonna draw some microscope slides. in
What
there.
about page 37?
What does that have on it? Oh, we're gonna do that next time. That's our lab next time.
No.
All right, it is lab time. What we're gonna do?
Wait,
We're gonna
is this
try
on
it-
lab?
No, dude,
No, that's
listen.
homework.
Wait, just the next.
The little 2 circles at the bottom of that page.
Oh, we're just drawing.
That's where you're gonna be lab. Yeah, we're gonna draw some microscope slides in there. Here's what we're gonna do. We're going to look at your cheek cells, but we have to get those ready. They should be dry on those slides by now, okay? And then the next thing we're gonna do is we're gonna take an onion. We're gonna peel a thin section of onion skin and put it on a slide, put a drop of iodine and a cover slip, and we're gonna look at it under the scope. If we have time, I have a banana that we can smear onto a slide. Maybe I'll just make one of those, and then let everybody look at it. We also have a piece of spinach to try to peel off a little bit of spinach and put that onto a slide.
Can we eat the onion?
No, you may not eat the
You
onion.
wanna start crying?
But you're gonna make this. It's called a wet mount, and I want you to do that yourself. What you're gonna do right now with your cheek is to do a dry mount. So take a look at your slide. Does it look pretty dry? Okay. So Hayden, you're gonna hold on to this for me.
I eat Skittles. They're quite good.
All right. There's something we have to do to your slide to make sure the sample stays on it. And that's something we're doing, I need y'all to listen closely to me, because we're using a flame, safety. We need to heat fix it onto your slide.
Yes.
So here's what's gonna happen. I'm gonna light this little candle.
First.
Okay, so, Troy, what you're gonna do is take your slide. Have you done this? No. Okay, so you know that the top is there. Make sure your sample's on the top. You're gonna pass it over this like this, just a little bit, and then touch it to your hand right here until the slide feels warm to the touch. Can you try
Afterwards,
that?
may I put the flame out with my fingers? With a
Maybe. So the bottom of your slide's gonna touch this flame, not too, don't get too close, 'cause it'll turn your slide black and maybe put the flame. So just pass it over it, and then touch it and make sure it's just warm. You don't wanna burn yourself.
Get too close.
Did it get, yeah, if you're too close, you get that black stuff on the bottom. It's okay, when you put it against your hand, it'll wipe right off. Did it feel warm to the touch? Okay, good. Now I want you to set it right here on the side of the
yeah,
stove.
that's the blunt.
Yeah, no. You're gonna set it right like this. And you two
Say
can
it
share
like
that,
that.
one-on-one fella on this. Once you have heat fixed your sample, you're gonna set it in the side of your beaker or cup, okay? And when I say you're not gonna put it in the top, Stephen, you heated it up nicely, it's gonna sit right there. Okay? You feel good?
Alright, so I'm gonna turn black in a minute.
It's okay to wipe off.
Okay. I don't like my slide being black.
Okay, here you go, you're gonna set it across here like this, just like this. Okay, hey, you come over with that bottle. This is my brand new methylene blue stain, the most famous biological stain out there, and I have a new one. So,
No,
everybody
I like
listen,
these
listen
things.
closely to what you're gonna do. Does everybody heat fix their slide? Eddie, have you done it yet? No, okay, you're probably getting it. And set it on the side of that cup there, don't let it go in. Once y'all are done, go ahead and blow the candle out before you catch somebody on fire.
Set it out with your fingers.
Alright, so then you're gonna take this stain, it's purple, and you're gonna do something we call flood the field. Alright, so Stephen, take this and drop it right to where your sample was on there, just nice little drop all over it, and then start counting to 60.
Okay.
Put another nice drop,
Mississippi to Mississippi.
and then connect them together with another drop. Okay, so count. They're gonna be, so right in there, like flood the field,
Okay.
and then you're gonna count to 60.
1, 2.
Okay? And then Stephen, when you get to 60, it doesn't have to be exact. You're gonna take the whole slide and dump it down into the water and go like this. Pumps up. Huh? Okay, hand it to Eddie. Now, make sure not to touch your slide with the tip of the stain. That'll ruin my stain bottle. Some of your cells will get into my stain and destroy it. You didn't touch it, did you? Let it drop. Flop. There you go. Now
You know
count
my,
to 60
my
or
last
so.
name means thing.
A few, 2 or 3. Just kind of cover up that whole spot that was there. You got a lot of cells there, so we should just, there you go. And it was rough. Okay, now, do you see what Stephen, he took the whole cell after 60 seconds, take the whole slide and just set it down in there and go and wash it off and set it back on the side of the cup. There you go. Perfect.
It's not really water.
Don't scrape it, don't scrape it. Okay, yours, go ahead now. You good or you still counting?
Yeah, we're good.
Let me do it for you.
And the next thing we're gonna do is our onion slide. Alright? Because we have to let that one sit there and dry. The amount of time I used to wait for my slides to dry.
It's way too complicated.
Okay, so we're gonna need another.
Like you can't feel it. It's not-
Wait, so did you dunk it?
You dunk it after 60 seconds or so.
I don't.
Okay, so I'm bringing you another slide. So what we're gonna do here is we're gonna peel off.
Have a little piece. It's that little like papery stuff.
Yeah,
Natural. Okay.
like that. Okay, then cut it a little piece and just set it right there on your slide. I'm gonna go ahead and write this on here. Okay, let me get another. I might not even need it. Okay guys, oh, I need to get you a slide.
What are we looking for?
It's
So pretty.
It's good, it's fine, it's been stained now, so just leave it there to dry. I'm bringing you another slide.
Does this taste like blue Fanta?
It is poisonous, so don't do that.
Oh,
Alright, peel this
Eddie,
little
drink
piece
it,
off
Eddie,
here
drink
and
it.
set there you go. Get a little piece of that onions, kind of really thin skin stuff.
Hurry
But
up,
would I
Max.
die?
Damn, Max.
But would I die if I drank it?
Not immediately.
I will cut your throat.
So it would
Slowly. Don't drink it.
okay.
You just slowly die.
Ready?
Okay, so we're gonna do
I
some
want to
onions.
deal with a bunch of
And I tell your mother you're gonna get it.
I'm gonna tell you what, do some voice on this.
So I want you to peel that off right there. Okay, get a piece like that and stick it right down on your slide like that. Okay, and then we're gonna stain this one
Here,
with,
use the scalpel.
Judy, scalpel, that'll help you a little bit. Here's some that's already coming
I
off.
took
Eddie,
the
right
whole
there,
sheet.
some. And then we're gonna take this iodine and we're gonna drop it onto your onion.
Here. Yeah.
I messed up.
Now, just a nice drop on your iodine, not too much. Okay, I need you to get everybody A. Yeah, it needs to be pretty flat. I need you to get a little cover slip for everybody and take and try to get them, they're made of glass, so be careful.
Okay.
Right, and try to separate it. See, like this one might be two, and so I'm gonna try to,
Where did you
if I can't separate
How
them, I
big
can't
is the
separate
drop sheet one? Is this big?
Very
Yeah, and then we're gonna set a cover slip down.
Are you going on that side?
If he
Set
stabs
it down,
me,
just
my
tap,
suit
tap,
can-
tap.
Yes.
Okay, now I want
You
you to
signed
put
the
that under
contract,
this
you
scope
can.
right here.
that-
So, okay.
Alright, who's got their, onion ready to go this way? Hayden, right over here. We're gonna just put a drop of this on. Go ahead, take a drop
You over here?
onto your onion.
Put our onion.
And then Hayden's gonna give you a cover slip and just put the cover slip right on the drop. Hand that over there. Yes, yeah.
Eddie, shut up. Eddie, shut
You
up.
see it? Okay, that's good enough. That's good
Yeah.
enough. Take this. Oops,
I did it on purpose.
you see it there, don't you? Okay, tap it down with your fingers. Oh, there's two now. Put it under this. Starts with this.
Here.
Come on. Yeah.
I gotta get this.
Did you get one too? Yeah.
All right, Eddie here. A drop of this, please. Try not to squirt it. It'll stain your shirt forever. That's what I have a stain on my coat here with that.
So I don't know
These
how,
are
but
Clorox
somehow the
paper.
onion got chopped. I don't know what happened, but I looked away and
I know
the
what
slice of the onion came off.
I know exactly what happened.
Max, so, Max.
Okay, once you put a drop of iodine over it, then put the cover slip on it. What's going on there? They're so
Would you
cool.
go in?
So just put a
Yeah,
couple drops
I'm
on
looking.
there, and then put the cover slip in the spot over the iodine, okay?
Oh, just go over. Oh.
Yeah,
okay.
cover slip goes over the iodine.
I have two cover slips.
Alright, Eddie, okay, can you bring that
Yes.
microscope, please?
Sure.
Here's the scope, put it under, take a look at it, draw it. And they've got one, put one down at this end, give one, Stephen has one, so yeah, y'all can have another one here.
Wait, which one's the waist one again?
The smallest one.
You actually stabbed me earlier.
Start at the smallest.
Yeah,
Alright,
you're
so
a hard
just
one.
put that in there.
There you go,
Yeah,
so
you're gonna
we're
say,
ready. I didn't find my
onion
costume.
skin for one of them, onion skin,
What?
and then you're gonna have cheek salve.
Okay.
Oh, I'm gonna use a little bit of this on banana.
Whoa.
You should see little squares like Mr. Hook saw.
Shut up, Max.
Alright, where did that blade go?
It's right here, man. Max is
Thank
with it.
you.
I'm over here just with the onion. We're gonna throw it in there.
Okay, hey, now let me make this slide. See that iodine right there? I'm just gonna smear this right here, buddy. And that little thing right there.
It's back in my clock with a scalpel.
Just smear it like
He's gonna
this.
cut my throat.
Go ahead, put some drops there on the banana.
How many?
No, 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 iodine.
Scalpel. He's the one to do it in the class section.
So just don't touch it to the slide, just drop it about it and go with a couple of just a little bit there.
The microscope. Oh.
Yeah.
You think easy way to do that?
And now go ahead and put the cover slip over,
Look
maybe
at that.
two
That's
covers.
cool looking.
This is one we frequently use for our original.
Move your head, move your head up.
Turn it on. I win.
Alright, you see your, cells there, and everybody saw how to do our onions, so I'm gonna turn this off because it's making this room hot.
Honestly, I'm just seeing yellow.
Honestly, I'm
Do you
seeing
see any
Spinning
boxes?
it like you're in your you're
Alright,
gonna break
there.
it.
There's like random white spots.
Okay,
I'm honestly
start
seeing a
here.
big
Start
forehead.
right
Wait,
here.
that's
Yeah,
nice.
Angel,
bring it in
maybe-
closer.
Have that knife back? Oh.
You see those squares?
Oh yeah.
Okay. So if you wanna go up another level, you can, but you don't need to. What do you need, Ben?
Let's see.
Do you
All
need
this.
something?
No, I know.
So I pass this around now, and
We need a microscope for that one, okay? So let's go ahead and see who's got one. This, okay, see
What?
how it's turning black?
Mm-hmm.
That's cause the iodine colors the,
I mean, that is also true, but.
That is
starch.
pretty
Right.
giving there.
And if I just got a pile of table.
On my side. Alright, wait, actually, I should have bought it.
It's alright, I can put it over.
I love those files like
I
that.
finally got it.
I see an onion.
Yeah,
I
not
think
me.
I see salt. That's
Max, why did you mouse it?
It looks so cool
On your slide.
when it's not under the
Like, it was
microphone
on it.
glass.
Yeah. Did you draw the picture then? It looked like that, you just drew the edge of it?
Oh, I'm not done yet.
Oh, okay, you're just gonna remember?
What'd you call me?
I'm gonna put this banana in real quick and see what it looks like. Ball, ball.
You can. You can try, you could. Yeah, I want you to try. Wait, no.
It's getting scared.
Oh, look, it's white. This is the, okay. Looks like it, bench more than 120 something.
Okay, so what this one shows, is this banana here? We're gonna try to look at, don't draw this one. See all the little dark spots in there? It turns out the iodine gets into the starch part of the banana, and those are little vacuoles holding starches. Little starch vacuoles. So just glance in there, Max, while you're waiting. That's banana. Alright, also check out these. Oh, why is that down in there? It needs to be drying, man.
No. Didn't know. What'd you get cut from?
It's actually a blister that just won't heat.
Oh, she got cut with a scalpel.
I've done it before, for sure.
So, do not drink the liquid
This
from my
one's
finger
yours,
right
the one
now.
that fell in.
What are we looking at?
Yours. Okay, could I get you to pick up all these blue cups and dump them into that bucket right there? We're gonna see if that
These
starts
draw
to
the
dry.
onion?
Yes, draw the, not the banana, but the onion.
Okay.
This is still the banana?
Yeah. Would you?
Eddie, take a look at this banana real quick. I wanna see those little black things, that is the little vacuoles that hold the, starch, and the iodine turned it black.
You report in this,
Just right in the bucket.
Okay.
That's kind of like our sink. And then rinse them out a little bit, rinse out those cups a little bit.
If you were on fire, does that
So you're not gonna draw that, this is the banana?
Yeah, that's how I was thinking.
Okay, you drew that, you don't have to draw
Ah,
that.
you're
We're
right.
gonna go ahead and now take this slide out of there and see if this one's dry. If your cheek slide isn't dry yet, we gotta wait for it to dry. You cannot put a wet slide under the microscope unless you have a special kind of a.
Hey, cut that scalpel back.
What?
Cut that scalpel.
You can wipe the bottom of your slide off, but do not wipe the top off. You'll wipe your sample right off.
Shit.
So what I'm gonna do is I'm gonna put this under the light right there. Right now. You're not gonna see it. It's just gonna help dry it. 'Cause there's a little bit of heat coming
I
out.
accidentally poured like a bunch on it.
Yeah, that's what you need to do. You know that's the top right here, top? So I'm gonna wipe the bottom off. But not the top, so yeah.
Ourselves to shame.
I get you? No.
I don't know how to do that.
And just use that to try to dry it.
You ever take this?
Yes.
Why does yours look just like mine? Eddie? No?
Alright, Stephen, you got yours under the, it's still drying? Okay. Yeah, if you wanna put the slide on the light of the microscope, you just look at this banana, y'all already looked at the banana?
But it's gonna be this.
Have we really had any recently?
Those little round black things are the little vacuoles that hold starch, and then the iodine exposes it.
It will expose
You
me.
turned Eddie, you turned Yeah, this your birthday party
No,
on
keep
purpose,
cut.
bro. No, I'm just messing. No, honestly though,
Alright.
it was gonna be, I wish I could go,
Okay,
it
that's
sounds
top.
fun.
Top.
Was it fun?
Yeah, now we have four.
I've been to the beach 4 times this year already. I'm going again next month. It's just boring now. Yeah, I didn't wanna go.
So I'm gonna put it right on this light right there.
White horses, I guess. Yeah,
You
with mom's
drew
red
your
horses on the beach.
onion.
Yeah.
Okay.
Thanks, slave.
That's
the table.
I can take care of that.
Miss Casper.
Is that your cheek?
Yes, ma'am. I already know
Okay.
that.
Hey, did you see anything?
No.
I want you to draw it. If you didn't see anything, let me try. Put it under and let me try to find it.
Is it dry?
I'm really good at.
Oh, the onion came off.
Judah, you ruined it.
Judah.
The
No,
onion-
don't touch it. Don't touch
Oh,
it.
God.
Why would you touch it? Don't stop touching
That's
it.
broken. It broke.
Did something break under the scope?
No, the glass broke on the corner of the thing, man.
You alright?
Yeah, I'm fine, yeah. The onion's here too. I'm
What's, how did it break? How did it break?
I tried grabbing it and I broke it.
Judah.
Break it.
Open.
Is the onion gonna
I
be
broke it by
okay?
massive.
Onions are alright.
I need to wipe it off.
Did it get on the objective?
No, man, we just got on this thing. Oh, it goes out?
Alright, where's your?
What? It's not my fault I broke it. Wait, actually, it's his fault I
You
broke
just
it.
said it's not my fault, I broke it. That
I
is
opened it.
science.
No, I opened it, but I grabbed the thing. I realized how
Yeah.
fragile it was. That's French. Okay.
It's not my fault that I broke it.
Isn't that French? Fried chili. I speak a little bit of French. I won't be that good.
Okay, so here's your cells, Judah. I'm gonna turn the light down a little bit. It's so bright.
Yeah, Max can hold it.
Oh.
So these are your cells. A little bit of blue in there.
So is the blue bacteria?
Miss Casper, mine's pretty dry.
Yeah, I need it.
Okay,
No, but
put it in the scope.
Okay.
Yeah, I broke it and I got well.
Take the old slide out. I'll take that. That's a piece of onion.
No, that's a banana.
Oh, that's the banana.
The banana.
Yeah, he needs
Everybody
it.
have their onion slides on?
What is it?
Get it.
He did.
So close.
He's saying he's the first.
Alright, so
This
doing
dye
that,
tastes good.
do you see your cheek cells?
Yes, ma'am.
Stephen, do
I
you
am
see your
not.
cheek cells?
Mine's still wet.
Alright, let's see.
What am I supposed to be drawing here?
That's right. I was waiting for someone to bring my check in, and now I have them this whole time.
What happened to this guy?
Alright, look for something blue first.
Okay, I wanna see mine.
So move the slide around till you see something blue, and then try to focus it, okay? Once you see something blue under there.
No, I'm just messing.
Okay, so now start opening those boxes, cause we're gonna put scopes in the boxes.
Okay. F. Scope F. Here's your bottles.
I'll see blue though.
Does it match up to the right one?
Yes, ma'am. What do you call them?
He said, here's your box.
Oh, they call me a microscope. Those them are quite a